My name's Larry Purcell. I'm a crop physiologist at the University of Arkansas. My research program mostly works with drought tolerance in soybean and looking at genotypes that can withstand drought better. One of the projects that I have funded by the Arkansas Soybean Promotion Board is looking for tools that can describe better drought tolerance in soybean. So we're looking at two basic methods of doing this. One is we are looking at color changes in soybean and how quickly they lose their greenness during a drought, during reproductive development, during seed filling period. The idea is that those soybeans that are sensitive to drought senesce more quickly and they lose their green color during the drought phenomenon. So we're actually going into the field and measuring greenness above a wide variety of soybean genotypes during seed fill under drought and under well-watered conditions. And we're following how quickly they lose that green color throughout the seed fill period. The way that we're measuring the greenness is we are attaching a camera to a kite and flying the kite over our soybean field for about 200 feet in the air. And from that height, we have geo-references on the ground with GPS coordinates. And so we know exactly where each plot is in the field. And we can correlate that with a genotype and then look at the greenness of the color over several different weeks to follow how that green color changes during drought stress. We're measuring the greenness values that we take from the air along with greenness values that we take from the ground level also to make sure our measurements are correct. The idea behind this is by making these measurements from the air, we can measure a lot more quickly and more efficiently a large number of soybean plots. Again, one of the things that we're looking at is being able to screen a large number of plants, large number of soybean genotypes at one time, and this gives us one mechanism of doing that so that we can follow their temperature during drought stress. This summer we were able to measure drought temperature responses in about 1,000 plots in less than an hour. So it's a very efficient means of doing this, and it's one of a very unique program that we have here in Arkansas. One of the tools that we're using now to identify these very favorable traits is molecular markers, and molecular markers are a big part of this program too. We can identify genes or chromosome segments that are associated with these specific traits, and then we can select those traits in a breeding program much more efficiently than previously in developing new cultivars. Eventually these cultivars are being folded back into the breeding program and they'll be available to farmers. This research would not be possible without the checkoff funding from the Arkansas Soybean Promotion Board. We're very thankful for the opportunity to work with the Promotion Board on this important research.